Uh, Welcome, everyone. Thank you very much for coming to my talk. My name is Ann. I'm a PhD student in Iowa State University, and I'm very excited to be talking to you today about my work, protein function, how much do we know and how much do we care. So I'm going to start with a well-known problem that the functions of proteins and genes are not fully characterized. We're all putting effort into studying genes and protein, and for some of them, we were able to call it a gene, yet we don't know what their functions are. Let's take the Human Proteome Project, for example, where their goal to set out is to find at least a function for every protein. They were able to answer the question, how many proteins are there in the human proteome? But now we're trying to find at least a function for every protein. How far are we until we reach that goal? Not only for human, but for other organisms as well. So I can tell you that the functions of these proteins are still unknown. 20% of human proteins, according to some estimates, or 35% of E. coli protein coding sequences, according to some estimate, or up to 60% of human gut microbiome coding sequences. This much percentage of protein coding sequences are still yet we don't know their function. So in the process of doing so, we realized that studies on protein functions are biased towards well-known proteins. So a little more proof, we show that the inequality follows a rich get richer pattern, which means that the annotation growth for the well-known protein is biased towards the ones that are richly annotated, a paper in 2018. And then more recently, we found out that the pace of functional discovery has slowed down, leaving around 20% of human proteins, even in well-studied model-like organisms, without informative description. And more recently, analysis of publication trends revealed that research efforts continue to focus on genes and proteins of known function. So we're paying a lot of attention to the known proteins, yet we are not paying enough attention to the rest of the proteins in the proteome, which leads me to our research goal. We want to analyze these biases in the study of human proteins over time, and we'll do that through two aspects. The first aspect, can you guess what it is? So it's knowledge, is how much do we know? And if these two pictures doesn't scream knowledge, I don't know what else will. So for anyone who doesn't know, these are two wise old men. The first one, Professor Dumbledore. I'm a fan of Harry Potter. And the second one is Gandalf. My PI is a fan of Lord of the Rings. So it only makes sense that I put both references here. So here it gets tricky, where my second aspect of when we want to analyze bias is interest. How much do we care? What other photos that I could put here? Now I have to use Hermione, who is interested in everything, and Gollum, who only cares about the ring. So it's a bit of a stretch of a joke, but yeah, the Gollum picture is minimized so that I don't want to jump scare anyone. Anyway, so this is going back to the title, knowledge, how much we know, and interest, how much we care. Let's start with how much we know. And here I'm quantifying knowledge of protein functions through gene ontology annotations. And I'm very sure that everyone knows about this, so I'm just gloss over it, but I have to include a slide about gene ontology. It's a standardized vocabulary of protein functions. This is gene ontology with the molecular function aspect and the biological process aspect. It's represented in directed acyclic graph, and since it is a graph, we need a metric to capture the information that we have from the gene ontology, which leads me to the information content. So the information content is what we use to quantify knowledge of proteins functions. Here I have two graphs for the term on the left and the term on the right. The information content was introduced by Claude Shannon in his information theory and first applied in bioinformatics in 2003 by Lord. And if you look at the term on the left and comparing it with the term on the right, the term in the red box on the left is more informative because it's assigned to fewer proteins. It appears less frequently than the term on the right, which is less informative, therefore lower information content. So to get the information content, we just take the minus log 2 of the frequency of that term in the corpus. And in my case, it's the human proteome. So generally, as we go down the gene ontology graph, the information content would increase. And the information content of the term increase. The protein that is annotated with more informative terms will have higher information content. So now we have a metric to compare the knowledge of protein functions. Let's see how that looks like in the human proteome. So here I'm showing a scatter plot for between information content in 2022 and term count in 2022. So each dot represents a protein, and the term count is just the number of terms that that protein was assigned to. So this scatter plot tells me that most proteins have low information content, where you see all of the black dots are basically focusing on the bottom of the y-axis. So that happens with molecular function and biological process. But this is, so we have like around 20,000 dots here. But if we actually go back in time, and here I'm collecting data in the Go annotations for the well-known proteins. And we can see the line is depicting the growth of these well-known proteins. And some remarkable growth is seen here with TP53 or TNF. So TP53, as we all know, the gene that encodes for tumor suppressor gene, or LRRK2, and PARK7 in green and red. Both of these genes have found to have mutations that is associated with Parkinson's disease, and TNF, tumor necrosis factor, linked to cerebral malaria or Alzheimer's disease. So the common thing about these highlighted proteins are they mostly associated with some diseases that we care about. That's why they grow a lot in information content. And you might notice that while their term count increases over time, their information content actually seems to saturate. So here I'm showing a box plot where I'm separating the well-known proteins that are highlighted in colored lines with the rest of the proteins in the boxes. So the high information content proteins kind of reach saturation where the lines have plateaued over time. And I have to put a secondary y-axis because it's like so much higher than the rest of the other proteins. But looking back at the other proteins, we see that the medium information content increases slightly over time from 2013 until now. But what consistent is the interquartile range, or how big the boxes are. They are big over time and doesn't seem to improve, which means that information content of these proteins vary broadly and consistently over time. So now we think we need another metric to quantify how much imbalance there is in this distribution of information content, which leads me to a metric that is commonly used in economics. The Gini coefficient is used to quantify the level of income inequality for different countries. So here's an example. We have Gini coefficients in 2019 for different countries, and the darker red indicate the higher Gini coefficient and higher inequality. If we actually put it as an overtime analysis, we have each line representing a country and their Gini coefficients over time. And anything above 0.4 is considered high inequality, which means there's like a significant inequality in that country. So now we adopt it to the same, do the same thing, but with our own metrics to quantify knowledge of protein functions. Here we have two lines, each for each aspect, and we can see that everything is above 0.4, which means there's high and persistent inequality in the distribution of information content, which means that in that the whole human proteome, there are a lot of proteins that have very high information content, and the rest of them don't have as much information content. But is that actually the whole story? In fact, if I divide this into a subset of proteins annotated in or before 2013, which means before the period that I'm looking at. So I can actually see a slight decrease in the red line, which means we are annotating the proteins that have appeared in the database for a long time in a more equal manner, which makes the Gini coefficient decrease a little bit, but only for a molecular function aspect. So the takeaway is earlier research proteins were annotated more evenly because they have spent a lot of time in the database and a lot of people have studied them. So that concludes my information content, or the how much we know part. Back to the how much we care part. How do we quantify the interest? So in my work, the interest is that proteins received is quantified through the number of publications. So now we're entirely switching gears from gene ontology annotations to the number of publications. And the interest that is quantified right now is used through total fractional count. Let's first start with fractional count. So this was taken from Jensen's lab. If you're here, thank you very much for updating and maintaining this database. So the fractional count is how many times each protein was mentioned, divided by the total number of mentions for all proteins. Let's go through a toy example because I have time. There's 3 publications. The first one, red, blue, and green. I'll go through one by one. For the first publication, it only mentions protein A, which means protein A has a whole fractional count of 1. But for the second publication, it mentions protein A and B equal number of times, which means it gives half to protein A and half to protein B. Similarly, for the third publication, mentioned protein A 3 times more than protein B, which means A got 7.5, 0.75, and B got 0.25. So the total fractional count is when we sum up across all the documents in the corpus that we're looking at. And that it will also be the total interest in this case. So protein A has 2.25, and unit is full publication equivalents, or FPEs, and B has 0.75 FPEs. Now we have a metric to quantify the interest. What I found is for across time, when I'm plotting the how much we care about proteins in 2013, and then the gain of how much we care, like the gain in interest over time, we're actually seeing a very strong positive correlation. What does that mean? The more frequently proteins were mentioned in 2013, the more publications that they gain over time. We're keeping studying the proteins that were heavily studied before, which means if we care more in the past, we'll probably care more in the future. So should I expect a same trend to the information content or the knowledge that we quantified before? It's actually not the case. This initial information content is not a good predictor of the gain in information content. So the same plotting the gain in information content with the initial information content, no correlation at all. Very low experience correlation seen in this scatter plot. What does that tell me? Why is that a different story from what we see from the number of publications? And we think that that might be a possible saturation of the information content. Remember the colored plateaued lines before? So some proteins already reach kind of their maximum information content. That's why we keep studying them, but they don't gain new annotations anymore. No new annotations mean no new information content. So they couldn't gain much anymore. If initial information content cannot predict the gain in information content, what would be the correlation between knowledge and interest? Or can we use the number of publications to predict the gain in information content because we annotate through publications? Again, I show a weak correlation between information content and the number of FPs over time. Here we see a lot of colored lines, but let's first focus on the bottom right of each plot, where I'm circling the proteins that might be familiar to you too. So those proteins were used as clinical assays. They're albumin, insulin, interleukin-6, or C-reactive proteins. They are mentioned a lot of times, but not for the goal of annotating new functions. They are just used as biochemical tools. They're not actual goals of functional studies. What about the rest of the proteins, which don't have a predominantly high number of FPEs? So the experience correlation between knowledge or quantified through information content and the number of FPEs is very low. Not very low, but it's not a very strong correlation. We cannot actually predict. But as we try to cherry pick, there are some rare correlations between knowledge and interest. And we found out that these proteins that are associated with these 4 diseases show a bit higher correlation between knowledge and interest. But if you look at closely at the Spearman's correlation, it's actually only 0.5 up to 0.58, which is also not strong at all. So what are the takeaways from this? The first one, we see a high inequality in the distribution of knowledge, which is quantified through information content among human proteins over time. We know that initial knowledge cannot predict the gain in knowledge, but the initial interest does predict the gain in interest as we keep studying the well-known proteins. So how much we know does not correlate with how much we care. The possible 2 reasons are the saturation in information content. Some proteins did not gain more information content over time, or a more important reason is we're gaining more knowledge, but this knowledge was missed in our work because it's not captured by the gene ontology, since we're only capturing knowledge through gene ontology annotations right now. So what are my future directions? I'll try to expand the quantification of knowledge of protein functions to include data from bigger sources that was not translatable into gene ontology, like tissue expression or disease association or human phenotype ontology. And maybe we can predict future information content of proteins and redirect the studies of protein functions to the proteins that with promising growth of information content. And with that, I would like to thank my PI who is here, Ido Friedberg, and the Department of Mathematics, as well as my program, for sponsoring me to be here today. And I'll be happy to take any questions.
Thank you. That was interesting. But I noticed a certain bias as to what you classified as well-known proteins, because I would have thought the housekeeping ones from the carbon cycle and the TCA cycle were the well-known proteins, and you didn't include any of those. I noticed a definite bias towards human and health, and a definite bias for some fascinating reason, the year 2013. Like gene ontology began in 2000.
Yeah, that's
what happened to the first 13 years? Right. So how do you classify your choice of well-known proteins?
Thank you for your question. Yeah, I agree that the well-known proteins in my definition are just the proteins that are having high information content. They accrue a lot of annotations. And what I use as my data in 2013 is actually everything that accrued before 2013. So we're just trying to look at the specific 10-year before data. But it doesn't mean that I'm like disregarding all the information in the gene ontologies 13 before 2013. So yeah, the well-known proteins is defined through information content. The ones that have highest information content is called well-known in our case. Does that answer your question?
It's an answer, but I disagree.
So how would you quantify the knowledge part?
Would you be able to ask that question like
Sorry.
earlier? So
Okay.
there's
All right.
another question ahead of you, but please go to the microphone.
Hi, really interesting talk. Some things I've been thinking about a lot. So thanks for doing the work and putting numbers to help me wrap my head around it. I'm really struck by your usage of the term saturation of information content. Because, I mean, theoretically, it's sort of pure theoretically, then information content could go up infinitely because it's a negative log, but also, obviously it can't because the gene ontology is only so big, and so you can only get some sort of whatever. I don't know what the smallest probability is, but also, again, biologically, we don't expect that. We don't really expect one protein to have a super unique pattern. And so when we see these curves topping out in saturation, what does that mean, right? Like, is that a result of the mathematical limitations of using the gene ontology? Is it a result of our experiments running out of things to find? Is it a result of any number of things? But also, is it even a problem, right? Like, because the information content is curving out doesn't mean that we're not still adding interesting, informative, functional annotations to, say, TP53. We're just adding more common ones now, but those still might be relevant for people studying TP53. So I don't know, I'm interested in your thoughts on that and why specifically you're using the term, saturation, as in the implications that it's sort of like running out and then there's no more.
Yeah, I agree with you
on
that using the word saturation is like kind of a bold statement. But in our case, it's saturate, meaning it's not increasing as exponentially anymore. It plateaued out, probably because we are running out of Go terms as of the gene ontology right now to annotate more functions. But as our technology improves, there might be new Go terms that added into the gene ontology, and TP53 could gain more knowledge. I don't know that. Or, but yeah, like you said, the knowledge that we are gaining for protein function is not entirely captured in the gene ontology. We're still learning new things about these well-known proteins, but since my knowledge is only quantified through gene ontology annotations, that is a caveat.
Thank you.
So thank you very much.
No, you have a question, or are you doing?
I have a question.
Oh,
Yes. Yes. So thank you very much for this talk. I have one very basic question, because you talked so much about biases. You rely on recognizing proteins in literature when you study the interest. How much is your work biased by differences in how easy it is to recognize protein names in literature? So some proteins might be very easily recognizable, and some proteins, they are very common acronyms, so they're very hard to, or very easy to mistake, so they're very difficult to recognize. Did you try to account for this, or find some way to test whether your work is biased by this?
Yeah, thank you for your question. The protein counts, I can easily tell that you can like contact Lars to figure it out, because like this is not my data, but thank you, Lars. But I am like trying to say that his algorithm, or like name entity recognition, is trying like its best to capture the gene and also their aliases and other stuff like that in the literature. So there's definitely a bias towards genes that are easily captured. And there's also cases where the gene names are kind of similar to each other, so it got messed up over there, but their protein identifier is entirely different. So that's also a naming inconsistency problem. But I would say that there is definitely bias in the protein counts itself. But we're trying to, what we're capturing is probably approximate count of how many times the proteins were mentioned in these publications.
Yeah, I'm just curious to get a dialogue going a little bit on what counts as a well-known protein. And I think there are various ways of trying to do that, right? So you are looking at ontology annotations. You could look at number of publications. There are more specialized things you could do, like look at textbooks or things like that, which would pick up the TCA cycle one. So Ido, do you want to comment on that?
Yeah, I'll just talk too much anyway, so I'll just try to be brief here. So the idea, what we tried to do here is basically look at the difference between, as Anne put it, interest and knowledge, and to see the interaction between them. Now, both metrics are not, because they're metrics, they're obviously not ideal, because they're not the real thing. They're metrics, they measure stuff. And knowledge, we only went by gene ontology, which of course doesn't capture all aspects. Knowledge, we went only by gene ontology, and the information content gene ontology, which of course doesn't capture everything. We could add HPO, like you said, that will capture the disease knowledge. We don't have that. If we add HPO, probably TP53 will not plateau, because we're adding new disease information TP53. All it really does in terms of molecular function is bind to DNA. So supposedly that's the plateau thing. The other issue that we have in terms of interest, that's not knowledge. We try to differentiate that. Interest is how many papers are written about this. It doesn't mean that if you write a lot of papers on a protein, that you know a lot about it. It just means that you write a lot of papers about it. And we try to and show that these things do not really correlate. And that's a really interesting finding. That's kind of, I think, the main thing here with Anne's work. So
Thank you.
that's what I tried to.
We're online listeners, so maybe you need to use the microphone. Go ahead.
Yeah, thank you. Great talk. Maybe just like a follow-up to that, and it kind of plays into some of the bias that was raised earlier. Really trying to wrap my head around the interest part, because you know, when you take into account the funding available to certain groups, and you know, the barrier to entry or activation energy to like do a study, right? And sometimes like this protein is like interesting right now, so right, someone's just like jumping on the bandwagon to do the research for it. How does that, I guess, maybe play into how you defined interest? Or someone just like throwing the name of the protein in their paper so it gets pinged, right?
Yeah.
Even though they weren't doing a ton of work, or it's more repetitive work. Like there's a lot of things around that I'm trying to incorporate with what you've shown today.
So there's definitely spikes, like you said, like some group just study a lot about these proteins and they find something interesting, and suddenly a lot of follow-up papers after that will mention that same protein. But what the corpus that Lars is using is pretty big. I think like I could say that those spikes could be insignificant over a long period of time and in a very big volume of documents. But yeah, there will definitely spikes. And also we're like not accounting for the money or the funding that goes into these proteins yet, which could be another metric that can quantify interest aside from the number of publications.
Yeah, I let the chat work because I wasn't as surprised to see that disconnect. Right.
Yeah.
I feel like it's so cool to actually see how you define like some of these metrics that really kind of gets those conversations going about these different biases in literature. So yeah, thank you.
Thank you.